get the slides up is to just kind of briefly go back and talk to you about how this emerged. How did we all get here today? And really, what are our goals for these next couple days? And so the RNDS community, the past, the present, and the future. And I think I advance. So it all started, as you learned last night, back in 1999, when we had the first Transverse Myelitis Symposium. And that was the first of its kind. It was really an awakening where people who had a rare disorder, transverse myelitis, got together. There were scientists there. There were physicians there. And otherwise I won't come out on the tape. And it was really empowering for the patients, I think. It was empowering for the caregivers, but it was also empowering for the scientists and physicians who were there. And it really spurred things to come in the future, which means this, I think, is very telling, that we have had now 14 symposia since 1999, whereas we had had 0 before that. And that tells of this kind of groundswell of support to study these, to look at these together, to look at the common immunologic mechanisms. And so let me go through a couple of these. So the first TMA International Symposium was in August of 1999, and it was followed in 2001 by our second one. And we decided at that point that every other year we would have it because of costs, both to put on the symposium, but also because of the costs of people coming to these. And I think that one was in Baltimore, right, in 2001? And Cody Unser, many of you know, has been a real dynamo for organizing awareness and education with transverse myelitis. And so she linked up with the Reeve Irvine Research Center in California in 2002, and we participated jointly in that. And it was a really fabulous conference, in part because the Reeve Irvine Center focuses more on traumatic spinal cord injury, and we focused on inflammatory injuries. But there was a real synergism as to how we started to think of those together. And that has led to an ongoing and developing relationship between Hopkins and University of California in Irvine. And I'll share that with you more later in the conference because we do a lot of our stem cell work together with the Irvine group. And that really was the initiation of that collaboration back in 2002. Chitra Krishnan and I went to Galway, Ireland in 2002, which was a fabulous trip, and we figured we just had to take one for the team and go to Ireland, and somebody's got to do it, and so we had a wonderful time. And anybody who was at this Columbus Transversemyelitis Workshop in 2003 will remember it as really an unbelievable event to see a bunch of kids there, some of whom were quadriplegic and ventilator dependent, playing, painting, doing arts and crafts. It was really a very emotional event. I'll tell you, one of the things that lives on is the Transverse Myelitis Association dedicated to Hopkins a huge painting done by the kids at that workshop, which sits in our outpatient center. And we go by it 20 times a day, and it's a real testament to how these kids with, in some cases, incredible disabilities, were bright, were positive, and it's just really an amazing piece of work. We expanded it to become the Rare Neuroimmunologic Disorders Symposium in August of 2004, and the idea was that there's a lot of commonality with other disorders like ADEM and Neuromyelitis Optica and optic neuritis and multiple sclerosis and sarcoidosis. And it's better for us, since each of those is a rare community, to study them together. And there are unique features of each, but there are also shared features of each. So to share resources, to share ideas, from the research side, from the clinical side, and the patient experience side made an awful lot of sense. And so that was the first one. The second one was in 2006. And then the Southwest Neuroimmunologic Symposium, sponsored by Cody and the Cody Unser First Step Foundation, was in April of 2007. We then had another very important event, which is the Victory Junction Gang Camp in the fall of 2006. And that's another one where if anybody saw that or was there, it is a real life-changing experience. And it was really fabulous. That first one was a long weekend where teens and older went to the camp and really met with each other and bonded with each other, and it was just a fabulous time. This, for those of you who don't know about Victory Junction, you've got to find out about it, because it is fabulous what they do. You'll hear about it from Dr. Peter Sim, but it's really a very important function. We tied on at the American Academy of Physiatry meeting another RNDS in 2007, London and Scotland TM group meetings in 2007, another one I had to take for the team. And it was just spectacular to see all of these people there and to be able to offer my insights. We had a full week, essentially, at Victory Junction in the summer of 2007 where families went, my family went, my daughters went, and they keep asking about it. They keep saying, we've got to go back, and it was really something. And then here we are in Seattle. So let me take you a little bit through a few-minute history of RNDS, or transverse myelitis, and I'm just going to kind of highlight some of what I think are the critical events that have happened in the medical literature. Bastien, back in 1881 was the first one to describe acute myelitis and described it as a vascular or immune-mediated attack. Rivers, actually at Hopkins in 1929, described some cases back in 1922 and 23 in the UK as post-vaccination cases. And there was an outbreak in the UK in 1922 and 1923. And one of the things that was very interesting is that on the continent, in Europe, they had done some similar vaccines, but the vaccines were made slightly differently, and there was not an epidemic on the European continent, and that led to some very important insights into how the immune system can be tricked into becoming abnormal and attacking self. The first diagnosis, the first term, acute transverse myelitis, as far as we can figure it, was back in 1948 by Suchet K, and it was used to describe a case of rapidly progressive paraparesis with a thoracic sensory level occurring as a post-infectious complication of pneumonia. And this is something that has continued throughout the literature. It typically occurs as the infection is going away, as the fever is resolving, as the pneumonia is improving, as the GI illness is improving, and that's really where the term post-infectious event came on. Dr. Berman in 1981 talked about incidence of transverse myelitis, and many of you have heard about the 1 to 4 new cases per million people per year. That came from this and some other studies, which kind of puts it as a very rare disease, and I suspect it's much more common than 1 to 4 new cases per million people, but that's where we have gotten that. Larry Steinman is a really good researcher who in 1983 talked about superantigens. Superantigens are little proteins or molecules present within certain infectious agents that trigger the immune system to go amok. And several different viruses and bacteria have evolved to encode these so as essentially to divert the immune system away. But one of the side effects of that diversion happens to be autoimmunity. And it's a very important concept that has become substantiated in the last 25 years, really since that observation was first made. Tippett in 1991 talked about relapsing transverse myelitis. Prior to that, we'd really thought of it as a monophasic disorder, one time come and gone, never to come again. But it turns out it's not so simple. There are relapsing cases, and we now know from O'Riordan's study in 1996 about longitudinally extensive TM, or Neuromyelitis Optica, which you will hear a lot about. Philippe Llorens came to those early TM symposia because he had shown a very important finding. In a patient with TM, he isolated a bacteria from an otitis media in this little baby. And he explored what happened, and it was a strep pyogenes. It was a wimpy little bacteria that occurred in this child prior to the onset of transverse myelitis. But as it turns out, this particular bacteria was really nasty, had a very specific superantigen, which triggered a massive upregulation of the immune system in this baby. And that was the first direct link of superantigens, which we knew kind of at a high level, but now was linked directly to transverse myelitis. Tom Scott, who's in the Pittsburgh area, has looked very carefully at if somebody gets transverse myelitis, what is the risk of it going on to become multiple sclerosis? And there are some very important clinical and spinal fluid and imaging features which can sort that risk out. It's a very important observation. And then Brian Weinschenker at Mayo did a wonderful study, which was to look at whether plasma exchange was helpful in patients with idiopathic inflammatory demyelinating disorders. And this was a very tough study for Brian to do because he did it the right way. He gave patients real plasma exchange or sham plasma exchange, and they didn't know which. But if he had not done that, we wouldn't know today that it is a useful therapy, and we now do know that. So that was a very important study, and the methodology is one which I think we can use as we go forward. Dean Wingerchuk and Brian Weinschenker published criteria for Neuromyelitis Optica. Jerome Desez, who many of you know because he came to one of the symposia, talked about what do you do if somebody presents with an acute myelopathy? And he focused on the very broad characteristics, which is it compressive or not? Is it associated with an infection or not? Those types of things. And that was important. I'll skip to the Transverse Myelitis Consortium Working Group because that was our effort to say, look, that first effort was wonderful. It told you how to think about somebody with an acute myelopathy, meaning an acute spinal cord dysfunction. But how do we really parse this out into different subcategories? And so you come up with an idiopathic transverse myelitis, a disease-associated transverse myelitis, that associated with neuromyelitis optica or multiple sclerosis, etc. And even more importantly, it provided a diagnostic and treatment algorithm. So it basically told physicians out in the community, this is how we would do it. If you see a patient with an acute spinal cord problem, think about this, do this, and if it looks like this, you go one way. If it goes like this, you go another way. And so that was the first time that that existed, and it has been modified really every year since 2002 as we have gained more and more information. Jerome Desez again came back and said, look, these guys put forth these criteria in 2002. We want to see how good they are. We want to see how they work. So we actually applied them and came up with a really good grouping classification for transverse myelitis and really came up with the conclusion that these criteria did work very well. They probably missed some patients with transverse myelitis because we were very rigorous with the criteria, but they were in general quite good. Adam Kaplan, who is here, published what I think is a very important paper in 2005 where he defined some of the cellular and molecular participants in transverse myelitis. Based on spinal fluid, he found out that there's a certain cytokine, he'll talk to you about it a little bit maybe, interleukin 6. So this is the first time we started to know what really is happening in and around the spinal cord. And he even worked out several of the other things that then get triggered by this interleukin 6, resulting in the spinal cord demyelination and axonal injury. Dean Wingerchuk revised his NMO criteria in 2006. Chitra Krishnan revised our treatment algorithm and put it online in up-to-date in 2007. So now any physician anywhere, a family practitioner, an ER doc, can go on up-to-date, which most physicians use. And really what we do is when we see a patient and we don't know what that patient has, we say, we'll be right back, and we go out of the room and we close the patient's door and we go to up-to-date, and then we look at what we think it might be, and we come back in and we act as if we're smart when we go back in and see the patients. So this is a very important resource that physicians use all the time to look smart. And to have this on there, I think, was really important, and it also takes into account the NMO data. Ben Greenberg, who is here, published a retrospective review of treatment options in transverse myelitis of, I forget how many patients Ben looked at, but 120 patients. And how did they do with various treatment regimens? if they got cyclophosphamide, if they got plasma exchange, if they got just steroids, etc. Frank Pidcock, who is here, published a really important review on pediatric TM, one that he now hopes to follow up with, as we've got more and more kids with transverse myelitis that we see. And Julius Birnbaum, who's here, is about to publish what I think is a very important manuscript linking lupus and transverse myelitis. He's got some very important data, and I think he's going to share that with you over the next couple days. So we'll go through this a little bit later, but this is the important kind of the diagnostic workup tree that now is available to physicians throughout the world online, and basically says, if this, then this, and tells you how to work down this to all of these different rare neuroimmunologic disorders. So what's missing? Why aren't we done with these disorders? Well, we've got to understand them more at a molecular and cellular level before we figure out how to treat them. Humans are very hard to experiment on, and animals don't fit the bill very well in many cases. We don't have, in some cases, animal models of these disorders. And once you do, things start to progress much more rapidly. The tools that we have in terms of modulating the immune system gone out of control are pretty blunt sometimes. Steroids globally alter the immune system. Many of the drugs that we have are pretty blunt tools, and we need things that are more precise. And then finally, TM was discovered in 1881. We've only really been at it since 1999. A lot of progress has been made, but we'll get further over the next 5 to 10 years. So what do we need? What are the steps that we hope to achieve and that this meeting is a part of getting there? Animal models of NMO, optic neuritis, transverse myelitis, etc. Cell culture models. Once we start to know the molecules, the cells, the proteins that are injuring the spinal cord or the optic nerve, we can now do this in cell culture. And the advantage of doing it in cell culture is we can start to do things very rapidly, and we can test new drugs. We need to understand how the events unfold. Not only what is the trigger, but what are the things that then really, in some patients, accelerate the disease, and then the final common pathways of neural injury. We've got to test new drugs. High-throughput screening is a way to start to test thousands of new potential drugs, and also rational design, which means we know the pathophysiology and we're going to select specific targets. And the key to this really is collaborative cross-pollination, researchers and scientists thinking and talking together. The clinical steps are multi-center consortia, decision on outcome measures. In other words, we've got to get together, and Sandy, this is one of his passions, put together groups of clinical centers that are going to think about how will the trial look? What will we define as success? That's the first step. The second step is to do the actual clinical trial, but you can't do it unless you've figured out what are the readouts going to be. How are you going to define success, and who will be enrolled? So we also need commercial investment, which with rare neuroimmunologic disorders is hard to come by. A specific rare neuroimmunologic disorder is not really commercially viable. There's too few people to make a big buck. So one of the ways that we have pitched, and I think starting to become more successful, is it can be a model of something that is commercially viable. You say, look, I know you don't care much about transverse myelitis, but it will get you in the door and may apply to multiple sclerosis. So that works. And they're doing it really as a stepping stone to a bigger disease, but we don't care. We want them to test it in transverse myelitis. We want to get through with these clinical trials. There is also an FDA regulation that says if you get a therapy for an orphan disease, the bars are lower. And so it gets you in the door more quickly. And so we've really tried to stimulate that for new therapy development in these rare neuroimmunologic disorders. So why study them together? Why are people with various disorders here today? Because a lot of the things that cause these injuries and propagate these injuries are shared. The trigger mechanisms, the T lymphocyte mechanisms, which you'll hear about, B lymphocyte mechanisms, trafficking into the nervous system, neural injury and neural regeneration are shared. And that's the rationale for doing this together rather than apart. So why have clinicians, researchers, patients, and caregivers here together? Because some magical transfer and advancement of knowledge and energy occurs when people of various walks of life are thrust together for a common purpose to eradicate these autoimmune disorders. So, don't talk to people you know. They're boring, and you'll talk about the same old crap. No question is too stupid. For every question asked of you, you've got to ask one back. And this is true of the researchers and the scientists, because we need to learn from you what has worked for you, what experiences you've had, what that we've told you has worked, and what has been just absolutely not true at all. We need to know this. And you need to believe that this will change the future for science, for you, and for the community. And that's it. So, let's get going.